Let's get started with today's lecture. Very conveniently, Lecture 7, Bacteriophage T7, and one guess what the triangulation number is for the virus? Yay! Woohoo! But that's not the clicker question for today. Sorry. Well, that will be for next time, right? So one for today is small, single-stranded RNA bacteriophages need cellular translation proteins to replicate properly because they, and they here refers to the cellular translation proteins. I just realized that looking at this question. Because these cellular translation proteins only bind to the negative strand, only bind to the positive strand, bind RNA, are GTPases, compete with the ribosome for template. No harm in guessing. You can always change your answer in the next 3 seconds. Okay, what did you think? C. 47% think C. The cellular translation proteins, at least the ones we talk about, the ones that are required for replication, clearly bind to RNA. They don't seem to know which template is which. Doesn't make a huge difference, although there is a slight difference between the positive and negative strands here. If you think about what do you need to make positive and negative strands? Do you need translation proteins to make negative strands or positive strands? To make negative strands. Positive strands, you just need the polymerase. So, but it's probably mostly because of binding the RNA, yes. They are GTPases, and potentially the G which gets added at the end may, again with a big question mark, be coming from one of these ATPases and competing with the ribosome for template. That's the reason why I went back and specified it was the proteins themselves, because if you think about it, the replicase is actually competing with the ribosome for these templates, which is why they is a really dangerous word to use in questions. So that's our first one. Second clicker question for today is the Phi X 174 genome sequence was thought to be a message from outer space because it was so small, the first genome to be sequenced, single-stranded, composed of overlapping open reading frames, very tolerant of mutation. ABCD. You guys are a lot quicker at answering this question.
Outer space.
Outer space. A message from outer space. You know, whether it came from outer space is a different story entirely. So is anyone else going to vote, or should we just stop? Give you another 30 seconds to sort of think about it. Get in the groove. We'll be talking about viruses from outer space tomorrow. Another reason to come. Potential viruses from outer space, anyway. Okay, what do we think? Yay, everybody overlapping open reading frames. Although all of these, of course, are true. It's just this, and these are the worst kinds of questions because it's the why question. It's because of this overlapping open reading frames. Certainly, very tolerant of mutation. It has to be because of the overlapping open reading frames. Yes, it's single-stranded. Yes, it's the first gene to be sequenced. And it is relatively small, but not the smallest of any of the genomes that we'll be talking about as far as the class is concerned. Okay, last clicker question for today. Microviruses are unique in their assembly in that they use RNA-binding scaffolding proteins, only one capsid protein, a procapsid, scaffolding proteins, internal and external scaffolding proteins. And again, you're welcome to change your answers. Okay, we're split about 50-50 between C and E. Having a procapsid and internal extra scaffolding proteins. Nobody likes RNA-binding scaffolding proteins. Maybe because they aren't RNA viruses. Might have something to do with it. Only one capsid protein. We've already talked about a number of viruses for which this is the case. A procapsid, which is that structure that forms before you end up with a final capsid structure. We have actually talked about one of those before. We'll talk about a lot more later. Scaffolding proteins, yes, there are lots of scaffolding proteins. Internal and external. That's the only one, actually, that we're going to talk about that has these two specific scaffolding proteins. Okay, so we can put away our clickers. Anyone needs to leave, bye. Come tomorrow to my science pub. So just real quick review for the microviruses and Phi X 174 being the example of that. And that is that we've got the genome, single-stranded, the structure of these microviruses, which is a nice T=1 icosahedral structure. Binding and entry, really binding again to the outside of the cell, and then the entry process we really don't actually know that much about. We'll know a lot more about entry of T7. The replication having to do with the nicking protein, which elongates, giving you rolling circle replication. Our assembly, again, with these two kinds of scaffolding proteins. And then I talked about some of the variable ones a little bit later on. And my computer here is telling me that the battery's about to die on my mic, and we want to be able to hear my recording, right? So I'll switch the batteries. Assuming I can actually get it open. This is always the problem with this thing. Oops. That looks better. Let me make sure that we're still recording here the way that we're supposed to be. Yes, it looks like we are. Good. Okay. Back to our microviruses, and actually now, again, Bacteriophage T7. Sort of the overview again here of phage T7. It's now a double-stranded DNA phage, so we did single-stranded RNA, single-stranded DNA, now the double-stranded DNA bacteriophages. Important aspects about Bacteriophage T7, it's still a relatively simple bacteriophage in terms of all the proteins which it encodes, and so still dependent on host, but much less so than some of the other phage that we've talked about so far. I wanted to again introduce the so-called T phages. Why are they called all of these T phages? And then entry for these guys is really kind of interesting, and it depends on the host transcriptional machinery to have genome entry take place, which is really interesting. And then the viral polymerases. We haven't talked about viral polymerases yet. Turns out that for Bacteriophage T7, these aspects of viral polymerases are really critical and have turned out to be incredibly useful for biotechnology, molecular biology purposes later on. Key concepts, again, from this lecture, a lot to do with transcription. So promoter cascades, the various different promoters which are involved here, particularly having to do with the bacteriophage-specific RNA polymerase, the T7 RNA polymerase. The whole concept of early, middle, and late genes. We're going to be covering this again and again and again as we move on through some of these other viruses. And then concatemers, which we talked about a little bit already. These are just genomes that are hooked up end to end to end, which then solve the end problem that you always have with a DNA genome, which is solved by Phi X 174 because it's just a circle. But any of these linear genomes that get packaged, you have to deal with the end replication. And the way a lot of bacteriophages deal with this is by just having multiple copies hooked up end to end. So let's talk about phage T7. Again, this is pretty standard kind of thing that we had before. Where does it come from? What's the structure? Binding and entry, transcription, replication, and biotechnology. The real keys here are going to be the binding and entry and transcription. This is a nice micrograph of a different bacteriophage. It's actually not Bacteriophage T7. It's one of the vast number of the so-called podophages, podo being foot, down here at the bottom. Basically, it's a tailless phage, but still has this structure at the end for binding and entering inside the cell. Where does Bacteriophage T7 come from? And in fact, all of the T bacteriophage, you probably heard about T even, T3, T5, T7, etc. These are all just type bacteriophages. These were chosen back even before the advent of the electron microscope based on their plaque morphology and different plaques that appeared on E. coli B and also specifically lytic bacteriophage. And the reason for that is it's a much easier cycle to think about than the lysogenic cycle, which is true for phage lambda that we'll be talking about on Wednesday. So these are just different kinds of plaques. Different viruses seem to be giving different kinds of plaques. That's how we're going to name them. So it was a really pretty straightforward technique. There's not necessarily any kind of relationship between these guys. Turns out that T3 is very similar to T7 and turns out to be really useful in terms of things like the RNA polymerase structurally. However, T1 and T5 are completely different. So that's one thing to bear in mind. We'll talk more about T4 later on in the class. Commercial phage preparation is where these things came from. Remember I mentioned phage therapy right towards the beginning, using the bacteriophage to kill the bacteria which are causing disease. So this particular Bacteriophage T7 came from one of these commercial phage preparations that was being used for treating bacterial disease. And again, not terribly surprising, it's infecting E. coli B. Where do you find E. coli? In the gut. So this was, again, where they came from in the first place. A lot of people use phage T7 because it's actually relatively simple relative to some of the other type phages, things like T4, T2, which have much, much, much bigger genomes. So it's a lot easier to do some of the mutagenesis, all these other studies. And much later, people found, again, these podoviruses with short feet at one end are very common in pathogens, which gets back to commercial phage preparation, but also just in general in seawater. And we'll look at some of those towards the end of today's lecture as well. As I mentioned as kind of an aside at the beginning today, Bacteriophage T7 has a T=7 head structure, and that's quite easy to see over here. It's a lot harder to see on the other image that I showed. So if we use the pen color red, start at one of the 5-fold axis of symmetry right here. Want to get to the next 5-fold axis of symmetry, which is over here. You need to go to 6-fold 1, 2, change direction, go over there 1. So gives you a T=7 symmetry. And then in this cartoon, again, it's really, hopefully, quite easy to see. One thing that is hopefully obvious as well is that down here at the bottom, even though this is a 5-fold axis of symmetry as far as the icosahedral head is concerned, this has how many legs? 1, 2, 3, 4, 5, and hard to see in the back, number 6. So for the tail structures and everything which is hanging off of that, this is almost always 6-fold symmetry. And so you have to have some connector between the 5-fold symmetry right here and the 6-fold symmetry down here. And that's through what's called this portal protein. And in this case, it's 12 subunits, but of course that's just 6 times 2. And it really serves as the adapter between this 5-fold axis of symmetry and the 6-fold axis, which is your tail, all of your tail fiber proteins, etc. I have labeled on here all of the names for each of these different things, GP10, GP8, GP11, GP12, blah, blah, blah. I wouldn't bother to try and remember all of them because I can't remember all of them, and there are, I think, 57, if I remember correctly, genes in the T7 genome. And so there's no way I can expect you to remember all of them. So I wouldn't bother. Anything these GP, et cetera, et cetera, et cetera. Important, however, is that you have a T=7 head made up of one protein, so quasi-equivalence in terms of how you put together that icosahedron. You've got a portal protein that, as we'll see, you find in lots and lots of different viruses. And then tail proteins with 6-fold symmetry. There are 6 of them. One of the things which is really different with Bacteriophage T7 and the close relatives, all the podophages, is that it has a very specific core, and we'll see this much more, and it's very important for how you get the genome inside the cell. What's inside that T=7 icosahedral head? It's a 40,000 base pair linear double-stranded DNA with some pretty unique ends, and you can measure it pretty easily. And again, getting back to the Phi X174, why were so many people interested in Phi X174? Because it's single-stranded DNA, so a great template for DNA polymerases. You can get lots of it. Here, double-stranded DNA, you can get lots of it. You can study it really quite easily. And so if you're interested in studying things that depend on double-stranded DNA, like transcription, it's a really, really nice tool to actually study. So what does that genome look like? It's got insane numbers of open reading frames, names which are completely incomprehensible. They make no sense whatsoever, unless you think about how they were made historically, or as one of my bosses used to say, hysterically. But basically, there are really these 3 classes of genes. And this is hopefully sound familiar when we talked about our one-step growth curves in terms of our early genes, or in some cases immediate early genes, our early genes sort of for the metabolism making more of the genome, and then our late genes, known here as class 3 genes, which are important for putting together the whole virus. So structural proteins here, late expression, replicating more of the genome here, the early or the middle genes, and then these very early protein encoding genes, which are important for regulating the host. And you remember the little bump that we had on that classic one-step growth curve where you see some of the viral RNAs pick up right at the beginning. Those are going to be from over here. So that's what that genome looks like. We'll look at how it's being regulated a little bit more later on. In order to get this genome inside the cell, you have to first bind the receptor for Bacteriophage T7 is a lipopolysaccharide. So LPS, very often, septic shock, for instance, very often caused by LPS. Classic extracellular molecule on E. coli. What happens? You have binding to the LPS, and then basically you drill your way through the membrane. What kind of bacteria is E. coli? Gram negative. What does that mean? How many members does it have? 2. So it's got to get through 2 membranes and a peptidoglycan layer. So it's got to get through these. Once you have a hole drilled into it, you release a little bit of the genome, which is really kind of interesting. It's only that tiny little piece of the genome, about almost 1,000 nucleotides of the 40,000 that comes in. Then the rest of the genome gets pulled into the cell by transcription, and the transcription is due to the cellular DNA-dependent RNA polymerase. So that pulls it inside the cell, and that means that in this first approximately 1,000 bases, that's how you get the genome inside the cell. So that's, in fact, what was known when this textbook was written a couple of years ago. More recently, we've actually learned a lot more about how the virus itself binds to the outside and gets that drill to go through in the first place. And so there was a really nice paper published by some of my colleagues in Science last year, a little over a year ago now, with electron tomography, which is similar to cryo-electron microscopy, slightly different. And basically, they took a bunch of grainy pictures, like you see at the top here, and made some really cool cartoons and movies out of it a little bit later on. But basically, what seems to be happening is you have these tail fibers, and this is just one of the 6, from Bacteriophage T7 that interacts with these external LPS, and basically the virus kind of walks around on the outside of the cell until it gets 6 of the tail fibers attached, and then you have this big conformational change where you've got this core set of subunits here, which basically sort of everts, spills its guts, kind of like starfish, ejecting their stomachs, where now you make a hole through the membrane, and now this is where your DNA can come on the outside. There's a really wonderful movie that they made of this, and so now I'm going to escape out of here quickly, and we'll take a look at the movie, which now, of course, has disappeared. This is the thing that you could see right at the beginning. Here we are. So it didn't like it over here. We'll look at over here. So this is Bacteriophage T7 reconstructed. And one of the really nice things that they were able to do in this study was look at viruses, or to say virions, that didn't have tails. You see there's no tails here. There's just this structure down here at the bottom. And this was great because what they could do was they could do comparisons between tailed virions and non-tailed virions in the electron microscope and really compare the two of them to each other. And so this is now that non-tailed mutant version with the capsid and the tail structure right here. That's that internal core, which we're going to be seeing released later. Again, 5-fold axis of symmetry down here at the bottom, but a 6-fold tail that's attached to it. This is what those tail fibers actually look like in this structure. And again, it's just a comparison between the mutant and the wild type with tails and without tails. And what they saw in their tomography were densities that were best modeled by each of these tail fiber proteins releasing from the capsid and interacting with the LPS on the outside of the cell. So here's this poor unsuspecting E. coli down here at the bottom. Here are a couple of T7 virions that interact with one, and apparently just one, of these tail fiber proteins, and then sort of walks around on the outside of the structure until you actually have some interactions with this tail spike in the right place. Then you have all 6 that interact. That leads to the conformational change, which releases the core, which can get through now the outer membrane, the peptidoglycan, and the inner membrane. They can even see indentations in the outer membrane when all 6 are bound. It's really cool. And then conformational change, where you have the, again, sort of eversion of what you had in here, and now you can see the bacteriophage DNA down at the bottom here. There are about 1,000 nucleotides, which is coming out of the bottom. My apologies, the link in the PowerPoint seems to be broken, but I'll make sure that gets updated and add it to D2L after class today. So that's binding. The cartoon now that we have in the textbook is here. Again, interactions on the outside of the cell. Now you get all 6 of your tail fibers. The core, sort of gray here, gets converted into basically a hole through the membrane. Now you have the cellular RNA polymerase, this green blob, which will transcribe, and in that transcription process is literally pulling the genome inside the cell. It does this until it makes enough of the next proteins in order to keep going. And of course, the most important of these next proteins is the bacteriophage RNA polymerase, the product of GP 1.0. No, again, horrible names here. But the T7 RNA polymerase is really fascinating for all kinds of different reasons. But probably the most important of them is that it's a single subunit RNA polymerase. And was the very first single subunit RNA polymerase to be found, at least DNA-dependent RNA polymerase. And this turns out to be incredibly useful for biotechnology purposes. It, just like all of the other DNA-dependent RNA polymerases, binds to promoter sequences. These promoter sequences are extremely different relative to the bacterial promoter sequences. See here from -17 to +6, where do bacterial RNA polymerases usually bind relative to the start site? Nobody remembers? So the -10, -35 sequences, the sigma sequence binds to. So again, very different relative to these sequences. Very different sequences, very different specificity now that you get. The other thing is that this polymerase cranks. It's about 6 times faster than the E. coli DNA-dependent RNA polymerase. So it can really pull the rest of the genome inside the cell. Yeah?
Is that T7 polymerase or the cells?
So the cellular polymerase pulls in the first little bit, and then once it's pulled in that first little bit, it's going to be transcribing. And that transcription, making the messenger RNA, in bacteria, you've got coupled transcription and translation. So the ribosomes will be then sitting on that first piece of DNA being transcribed into RNA, making the first viral proteins. And one of those very first viral proteins is this T7 RNA polymerase. So you get some entry due to transcription by the cellular polymerase, but much more once you get this polymerase in. And so the proteins now which are being made in the first 5 to 10 minutes are these immediate or the very early genes, class 1 genes as they're called in the Bacteriophage T7 literature. And we've already talked about our T7 specific RNA polymerase. There are a couple of other proteins which are encoded in these very early genes. Basically, they're regulating host functions. And so whenever we talk about these immediate early genes, the very early genes, the first genes that are being expressed, they're going to be regulating cellular functions. So one of those is inhibiting the E. coli RNA polymerase, which makes a lot of sense because as far as the virus is concerned, once it's infected, you don't want to bother with any of those silly E. coli genes. And one of the host defense mechanisms, restriction endonucleases. So normally, restriction endonucleases will chop up double-stranded DNA that's not been methylated, that's not appropriate for the cell. It's a viral defense mechanism. Well, one way to deal with that is to inhibit this. And you want to do this very early on, before too much of your genome has been pulled in due to transcription. So our immediate early genes are regulating host functions, but also one very important viral function. The genes that are made then by messenger RNAs made by the T7 specific RNA polymerase are now going to be the classic early genes, replication genes. I don't expect you to remember all of these. That's why I put a couple of circles on things. And again, I don't expect you to remember the names, but more the specific activities of these individual proteins. DNA polymerase. So it turns out this particular virus has its own DNA polymerase as well as its own RNA polymerase. There's DNA ligases, etc. Endonuclease and a lysozyme, which is really quite important. We'll talk more about the lysozyme a little bit later on. How do you make these genes that are important for making more of the genome? Well, not surprisingly, they're all made by the T7 specific DNA-dependent RNA polymerase that binds to very specific promoters. There's about 10 of these promoters spread out through the genome here. All of these genes up here, the early genes, are made from a couple of promoters that are in that first 850 base pairs. Those are the cellular promoters. So these are all making things that are regulating host function. And of course, gene 1 right here, that's the T7 DNA-dependent RNA polymerase. All of these transcripts through here, so I should row them over here so you can actually see them. All these transcripts are being made from the bacteriophage specific promoter. So there are sequences in the DNA and the genome up here which bind to the T7 RNA polymerase. You have a little bit of what's called terminator read-through. So all of these guys end up stopping at pretty much the same place, but they're starting at different places. That means you have most of the messenger RNA for these proteins down here. These proteins, as we'll see in just a second, are those proteins that are important for structure, scaffolding, and putting together the whole virus genome. Yeah?
So the first chunk up there is the polymerase?
The first chunks of these early genes up to TE? Yeah, so these include the polymerase. So these are up the top of these gene numbers. So gene 1, that's the polymerase. So it's encoded here in this little piece of DNA right there. 0.3 and 0.7 are the regulatory proteins for the host polymerase and the restriction endonuclease. But those are all made by the cellular RNA polymerase here. You also have a lot of RNA processing that happens in these messenger RNAs. You'll see all the vertical lines here. Each of those vertical lines represents a spot where the messenger RNA is chopped into a little smaller piece. The reason for that is actually to give these RNAs stability, which seems kind of strange. You chop them into smaller pieces, it gives you more stability. But that's because if you think about the E. coli messenger RNAs, usually those have signals, particularly at their 3' ends, which are important for those RNAs actually getting degraded relatively rapidly. Most RNAs in E. coli are pretty unstable. And so this process of chopping up these RNAs stabilizes the viral RNAs. What are those genes that you need lots of? Not surprisingly, they're scaffolding proteins, capsid proteins. If we back up here real quick, if you look at these genes right here, 8, 9, and 10. 8, 9, and 10 are those that you have the most transcripts for, which is not surprisingly also where you're going to be getting the most proteins from. These are your capsid protein. Remember, gene 10, one protein, everything that you need for your capsid. Scaffolding proteins here, portal protein. And then the last one of these down here that I really wanted to emphasize is gene 17.5. Again, I can't ever remember what 17.5 is. But it encodes a gene called holin. And holin does exactly what you expect. It makes a hole in the membrane of the host, which allows the completed virions to come out at the very end. How do you make these particularly class 3 genes, and how do you make more of them? Turns out you make them very quickly, but if you look at this genome cartoon, you'll notice that these lines are a bit thicker down here for the class 3 genes, and that's because the promoters have a closer to consensus sequence. And so the consensus sequence, that's the best binding site for the RNA polymerase. And so you're going to get more RNA polymerase binding to these promoters here than you get binding to these promoters here. Well, how do you get any of these promoters ever being used at all? Well, that's because it takes a while before this DNA is even inside the cell, before the viral polymerase can come down and bind to them and transcribe them. So that's one thing. The other thing, and I mentioned before, is the T7 lysozyme. The T7 lysozyme does what a lysozyme would normally do, which is break down the peptidoglycan, allow the virus to escape from the cell. But it also has a secondary role in T7 and in T3, the related bacteriophages, and that it inhibits the T7 RNA polymerase. And it turns out this is really important for the applications of using T7 RNA polymerase later, but also shuts down the production of a lot of these class 2 genes. So all of these class 2 genes here, A, you're not going to be making as much of them because they don't have as good binding sites. B, the amount of T7 RNA polymerase you have is going to be limited, so it's mostly going to be binding to these class 3 promoters down here at the bottom. So a couple of reasons for that. So that's how you're making all your proteins. How do you make more genome? Well, it turns out that with genome replication, as I mentioned, double-stranded DNA is really good for studying things like transcription, which you need double-stranded DNA for, but it's also really good for studying things like replication. Again, double-stranded DNA, cellular, what you need to actually function. So you need a DNA polymerase. DNA polymerases have one really big problem, and that problem is what? All DNA polymerases need primers. So you have to have a primer. Where does that primer come from? An RNA. In cellular systems, that's usually a primase. But in the case of this particular viral system, it uses the T7 DNA-dependent RNA polymerase to make primers. So you're kind of doing double duty, making messenger RNAs, but also making primers. So you have primer formation by the viral polymerase, and then, again, as I mentioned before, you have a viral DNA polymerase, which will extend that primer. There's a helicase as well. This helicase does just what helicases do, pull the 2 strands apart. Also has some primase activity, which will give you primers. But the first primer is always made by the T7 RNA polymerase. Otherwise, it's identical to what you would see in all other cellular systems: discontinuous replication, Okazaki fragments, etc. So, in fact, a lot of what we know about replication and these Okazaki fragments comes from virus-infected cells, where you're getting exactly these things that are taking place. The origin of replication is towards the left hand, or the early end of the genome. Why would that be? Okay, it's what comes into the cell first, but you're going to want to replicate the whole thing. So that's potentially one reason. But what was one of the big problems that we had with the RNA bacteriophage? We had the replicase and translation that were running into each other. Well, here, you're doing multiple different strands. So this polymerase, DNA polymerase, is going to be going the opposite direction to what? This would be where our transcription is going in the opposite direction. So these guys would be running into each other. And so here, if you're doing your leading and lagging strands towards the left end of the genome, the cell's not transcribing any of this anymore. And so you've got the freedom, as it were. You're not going to be competing with any of the transcriptional apparatus for doing replication as well. So now we've made our genomes. This genome is made as a single copy all the way out to the end, but still, because we need primers, we're going to have exactly the same problem that we have with all of our chromosomes, and that is that the primer is removed. We're not going to have RNA being inherited to the next generation. And so somehow you've got to deal with the ends of your genome. Now, the way that the ends of the genome are dealt with Bacteriophage T7 is that it hooks a whole bunch of genomes together. How that process happens is a very convoluted homologous recombination. We're not going to get into the details. But basically, you just hook up one genome after the other, head to tail to head to tail to head to tail to head to tail. And then those need to get packaged into the capsid proteins. Well, how does that happen? Well, it goes through the portal protein. So that adapter, which is going from a 5-fold axis of symmetry to the 6-fold symmetric tail, is in many cases, including T7, but we'll see in many other cases as well, it's an ATPase. It's basically a motor which packages the genome, stuffs the genome inside the capsid structure, inside the head. Once it gets inserted just one complete genome. Then there's a cleavage. This is an endonuclease cleavage at exactly one genome length. That's a full head structure. That then moves. You go to the next head, etc. And we'll see this for lambda when we talk about lambda on Wednesday. There's a nice animation of that as well. The scaffolded prohead structure, very similar to the case that we have with the Phi X 174, only it's just an internal scaffold. It's not an external scaffold. It's just a single scaffolding protein. We already talked about the holon. You make the holon, makes a hole in the membrane. That allows the virions to be released. That hole just goes through membranes. You remember there's also the peptidoglycan layer, which is there. The T7 lysozyme breaks that down in order to be able to release virus particles. So that's how you make more virions. They go off, they find a new E. coli, and they infect. Why are we so excited about Bacteriophage T7 other than the wonderful model in making more viruses? And that's because of the proteins, particularly the T7 RNA polymerase. So the T7 RNA polymerase is wonderful because it's a single subunit, so it's really easy to make, lots and lots and lots of it. Also has a very specific promoter sequence, very unlike any of the cellular promoters. So it's really, really useful for making a lot of your favorite protein, but also for making proteins that are otherwise really toxic to E. coli. And it turns out that a lot of proteins that we care about, that we want to make in E. coli, like human insulin or human growth factor, any of these things, they're actually pretty toxic to E. coli. So E. coli doesn't like to make them very much. And so if you just put the gene for your favorite protein into E. coli, and E. coli doesn't like it very much, then your E. coli is going to be really sick. If your E. coli is not sick, it's not going to make many more E. coli, and so you're not going to get much of your own favorite protein. So using the T7 RNA polymerase is a really great way to get expression of your favorite protein only under exactly the right conditions. And so basically what you do is you grow lots and lots of your E. coli, and then give that E. coli some signal to say either make the RNA polymerase from T7, and that's what's shown here in this slide up at the top. IPTG is used to express the T7 RNA polymerase gene. If you want to control that really well, you also make sure you've got some of the T7 lysozyme around. What does T7 lysozyme do as far as the polymerase is concerned? Blocks its activity. And so you only, when you get enough of your T7 RNA polymerase, are you going to start to transcribe your favorite gene, in this case gene X. Once you've made the messenger RNA, the cellular ribosomes will then make lots of copies of your gene X protein. In some cases, it turns out that even having a small amount of the T7 RNA polymerase around and a promoter for gene X is a problem. So you can actually even bring in the gene encoding your T7 RNA polymerase on a completely different genetic unit itself, like another bacteriophage. And so if you've got really, really toxic proteins, what you can use is a bacteriophage infection, recombinant bacteriophage, that has the T7 RNA polymerase gene on that bacteriophage. And this is what I had to do for one of the proteins that I used in graduate school, was to infect our cell with a recombinant bacteriophage that carried that T7 RNA polymerase gene. So that's one aspect of biotechnology. A lot of people use these so-called PET vectors. The PET vectors were developed to take advantage of this highly specific binding site for the promoter for RNA polymerase, and again, particularly for expression of toxic proteins, but just for large amounts of expression of lots of proteins, because it's a really fast polymerase, makes a whole bunch of messenger RNA, which can then get transcribed. Other aspects of T7 which are really useful for molecular biology purposes, we mentioned right at the beginning that there's the cellular defense mechanism against a lot of these double-stranded bacteriophage, which are restriction endonucleases. Bacteriophage T7 gene 0.3 inhibits these, and it turns out they didn't know at the time. They just had a mutant in this gene that then no longer was going to infect as well as the wild type, and then through lots of good genetic deduction figured out eventually that there were these restriction endonucleases. And of course, restriction endonucleases are critical for doing any kind of recombinant DNA work, because you cut your favorite piece of DNA with your favorite restriction endonuclease and put these guys back together. What this particular protein does is inactivates the restriction endonuclease, and in some cases, actually not in Bacteriophage T7, there's the methylase protein. So it's how do you protect your own DNA against getting chopped up by restriction endonucleases. You methylate your own DNA. Turns out that some of these viruses then block the methylase as well. And so it turns out that the virus infection process, when the cell tries to make more of its own DNA, it actually chops up its own DNA because it's got all these restriction endonucleases in the cell. And the virus genomes, some of them have actually evolved to not have any sites for restriction endonucleases in them, particularly for some of these smaller genomes. So really a fascinating process. And in the textbook, they also mention that it was this process that Luria and Bertani were studying in order to figure out restriction endonucleases in the first place. So I want to spend the last couple of minutes talking about some of the other podoviruses which are out there. As I mentioned before, this was originally from the T7 anyway, from a commercial preparation used to treat diseases. This is a publication from now 2 years ago, which infects Clostridium perfringens, which is usually for a food poisoning issue. And so the idea here is you could potentially use phage therapy to treat either the foods or potentially also disease. These are the virions themselves. Up at the top, you can see some of the tail fibers really quite nicely of one of these virus particles. So podoviruses infecting lots of pathogens. Another place that you find lots of podoviruses is in the oceans. And this was a publication from last year, together with a group down at Oregon State, Steve Giovannoni's lab, and together with Matt Sullivan, who's at the University of Arizona. They first saw lots of these dots, little yellow-green dots because of all the binding. But then they were looking for viruses that infected this bacterium called SAR11. SAR11 is one of the most abundant bacteria probably on the planet, particularly because they're in the oceans. For a long time, people thought there were no viruses that infected this organism because it was so small. This is, you can see here, this is a 200 nanometer scale bar down here at the bottom. This is just one of these poor SAR11s, which has just gotten completely packed with the viruses which are infecting it. So they found lots of these bacteriophage particles. SAR11 also has a very, very small genome. It's only about 1.8 million base pairs in length. So also people thought that meant no viruses. No, we can infect SAR11 and find all these different viruses. Was again published in Nature of last year. One of the things that was also done with this is to look at the structure of one of these podoviruses. Hopefully it's also really easy to see here that we're going from 5-fold axis of symmetry to 5-fold axis of symmetry. Here we've got another T equals 7 particle, but most important is this structure down here at the bottom. Again, portal protein with an internal core, and that internal core everts down here, and they call it the nozzle down at the bottom. He's at the red structure up here, becomes the end of the purple structure down here, probably in a manner very, very similar to that animation that I showed right at the beginning. And so these podoviruses are probably replicating in an extremely similar fashion to Bacteriophage T7 and Bacteriophage T3, a lot of the other podoviruses which are present out in the oceans. Probably, although we don't know, that's just going to release a small amount of the DNA here, which then cellular polymerases will pull through. Then there's a viral polymerase, which will then transcribe and pull the rest of the genome in. We don't know that for many of these podoviruses. We're just predicting that that's going to be the case. But certainly in terms of the structures are extremely similar relative to each other. Last thing that I wanted to mention as far as Bacteriophage T7 is this figure here on the right-hand side, where if you just look at the proteins that are being produced by a particular virus-infected cell, a couple of important things here. We've got a control. This is just all the proteins which are being produced by the E. coli. 0 time, right, when you start the infection, all the same proteins. But just within a couple of minutes, literally 4 to 6 minutes, you start to see some very strong expression of viral proteins. This one up here, protein 1, what's that? GP1 is the polymerase. So you see it's being expressed early on, but then goes down. My pointer here is disappearing on me. So expressed early on, then disappears because it's only being expressed from that messenger RNA, which is being made by the cellular RNA polymerase. That gets shut down by some of the other proteins you make, but you only need an enzymatic amount of it, so you don't need that much. On the other hand, these proteins, particularly 10, 9, and 6 down at the bottom, these are our late proteins. We're making really massive amounts of them. These, of course, are your structural proteins, which you're going to need to put this whole thing together. This process is incredibly reproducible, and so people are using it now for simulations and trying to model exactly how transcriptional cascades and transcriptional regulation leads to more complex phenotypes. But at least in this case, we know exactly how those are happening. Yeah?
So that was the RNA, is what that's on that gel, or is that the protein itself?
So these are all proteins here. So this is just proteins.
So why wouldn't the ones that are created actually just stay as it continued through? Because theoretically, aren't a lot of those
So the reason for that, so this is a great question. So the question is, why don't you still see lots of, for instance, the T7 RNA polymerase? Why don't you see lots of it over here? The reason for that is that the way that this experiment was done was loading the same amount of protein into each lane. And since now you've got so much more of things like the 10, 9, and 6 gene products down here, just the relative amounts of these are going to be so much more. And so you're actually loading less. You see there's still some of this protein, and if you follow it all the way out here, there'll still be enough of that, and it's plenty. So you don't actually see a degradation of that protein. It's just less which is being loaded on the gel. And that's, you can see actually a little bit better here when they're doing the modeling. On the other side is, yes, you've got this protein all the way through here, relatively speaking. So unlike in eukaryotic systems where you've got all the degradation processes, ubiquitin, et cetera, you don't have that in these cases. Okay, so that's it for T7.